Hi everyone, my name's Kat. I'm a research fellow at the University of Auckland working with Anna and Annabelle, and for my postdoc I'm working on the role of T's in invasion. But what I'll be talking about today, I think my title slide has disappeared, there we go. What I'll be talking about today is actually some of my PhD work. I focused for my PhD on the invasive European starling as mentioned, and this is when I revisited that story, the story of genetic diversity and selection using structural variants. Now, this was chronologically the last bit of my PhD that I was working on, so my head would not be still on my shoulders today, if not for the amazing undying support of my supervisors, including Lee right there in the back. And also, I'd like to thank all the funding I got, both for the project itself and for being here today. So the research that I do is primarily in the fundamental space, using invasive populations to look at evolution. Because when an invasive population is brought from their native range to somewhere new, they're bombarded by a new selection regime and undergo a process that I'm sure you're all very familiar with, which is rapid evolution. This all happens on a backdrop of fairly minimal genetic diversity and over a short biological time frame, which means that if we observe these populations, we can look at evolution happening in real time. And if we use omics approaches to do so, we can look at the fundamental molecular mechanisms that facilitate adaptation. Primarily what we use in the invasion genomic space at the moment is reduced representation sequencing data, which allows us to get at SNPs and indels. But slowly we are seeing trickling in whole genome resequencing, which I understand is completely not feasible for some organisms. Their genomes are too rude, they're messy, big. Sometimes we haven't even got the genome yet. But for the organisms that we do have whole genome resequencing data for, it's absolutely vital that we use this data to observe beyond the SNPs, beyond the small indels, into complicated genetic variants such as structural variation. And that's exactly what I wanted to do for my last thesis project. And I was so excited before realizing exactly what I'd gotten myself into. So, brief complaining session. Structural variants are really, really annoying. Here we have the reference individual in blue, the resequenced individual in green, and we see a stretch, the red stretch of DNA that's not in the reference. Now, how can we map this from the resequenced individual to the reference genome? It sounds reasonable if I tell you that's 30 base pairs long and our resequenced reads are short reads that are 150 or 250 base pairs long. But what if that's 1,000 base pairs long or 10,000 or even more? It gets really messy when we also realize that structural variants really like occurring in hard to map regions of the genome where there's high repeat content. And this is just one type of structural variants. There are so many more, and each of these require their own unique set of criteria to support their existence. Even if we have a perfect data set, because a philanthropic billionaire has given us lots of money, and we have a pangenome made of long reads, and we've got all our resequenced individuals done with long reads as well, our data set will still have a depressing amount of false positives and negatives in them. That being said, despite all this complaining, it's really important that we do study structural variants where we can. Because, for instance, in humans, the SNPs only make up 10% of the inter-individual variation. And across the species where we have begun to characterize structural variants, the total length of base pair changes introduced by these structural variants outlap many times over that of SNPs. And so it's important, if we want to understand how species evolve, we need to understand standing genetic variation, and structural variants make up a huge component of this. So to quickly touch back on the story of the starlings, they're native to the Palearctic. They were brought from the UK to basically every other continent, including Australia. We have a continuous range of starlings across Australia covering the southern eastern portion of it, and across which we have 5 distinct geographical introduction sites. We have fairly accurate historical records that give us not only the dates of first introduction, but the dates of the repeated introduction, because they were doing this deliberately. And we know where these birds were sourced from. Most of the time they were sourced from the native range, but sometimes not. I'll come back to this later. We have mitochondrial, microsatellite, and reduced representation data breaking up the continuous range into 2 major subpopulations, which are largely a result of differences in founder individual genetics, likely coupled with drift and selection as these populations expanded from the introduction points. And I'd also draw your eyes to the red section on the left there. This represents Munglanup, or the westernmost range edge of the Australian population. It's a site of highly reduced genetic diversity, completely unique haplotypes that we haven't seen anywhere else. It's thought that this is a result of selection and bottlenecking at this westernmost range edge. Overall, in Australia, the genetic diversity is much lower than that of the native range. This is to be expected because only a subset of individuals were brought over. But interestingly, the genetic diversity in Australia is also lower than that of North America. And the reason this is surprising is because the North American introduction only had a singular geographic introduction site, and less individuals in total were brought over. And so we wanted to revisit this story of population genetics and then selection within Australia using structural variants. So to do so, we had 49 whole genomes, 33 in Australia and 8 each from the native range UK and North America, and we contrasted the patterns of SNPs and structural variants. Looking at the SNP data summarized in PCA, we see the relative differences between the populations there, and Munglanup, the sort of aqua population there, sticks out like a sore thumb. And this happens also in the structural variant data set. Looking at both these types of genetic variants, we see such a huge amount of private alleles in this Munglanup range edge population that we're starting to suspect that maybe this genetic differentiation is not just due to genetic bottlenecking and selection, but perhaps there have been sneaky alleles introduced into the population from unsampled individuals that were somehow introduced to the western coast of Australia. And because these, in some cases, haplotypes haven't been seen anywhere else where we've sampled in the range, this must be an area that we haven't yet done any genetic work on. I'd also like to draw your eyes to the barely visible light blue dots there, which represent Hobart. I mentioned earlier that some sites were repeat introductions, and some of those introductions were sourced from not the native range, and Hobart is the worst offender for this. We brought over a whole bunch of individuals actually from New Zealand to Hobart. And so Hobart and Munglanup both represent sites where likely there was admixture, mixing of two genetically distinct populations, and we see them sort of pulled out in the PCA here. Despite the fact that in structural variant data sets, there is a great difficulty in not only determining the existence of a structural variant and then genotyping the individual, it's comforting to see that the relative patterns of structural variants and SNPs are fairly similar in these plots. And that's even more so the case when we do admixture analysis. We can see, despite the fact we're using hundreds of thousands of SNPs compared to only about 10,000 or so structural variants, which usually are a lot more dodgy, we see the admixture plot is basically identical. Where things start to fall apart a little bit more is when we look at genetic diversity. So comparing the SNPs on the left to the structural variants on the right, what jumps out at us is that the relative diversity of structural variants in the native range in North America are much lower compared to the rest of Australia. And in particular, we see the westernmost range edge Munglanup and Hobart have increased structural variant genetic diversity relative to SNPs. So what this is maybe suggesting to us is perhaps something about complex introduction histories, and specifically admixture, is having a disproportionate effect on the genetic diversity of structural variants compared to SNPs. Maybe. What this slide is missing is a giant asterisk right at the end of that. That is all the technical difficulties of working with structural variants. And I currently have a big laundry list of caveats. I'm cutting the data differently, refiltering it, and I actually haven't told you, Lee, but I'm recalling basically the entire structural variant data set against the North American genome, just to see whether or not these patterns stick, but so far it has. And so at least for the structural variants, we've been able to call with our short read data, which is highly biased towards deletions because those are a little bit easier to identify. This pattern seems to be holding true. With the 10,000 or so structural variants we had a quick look at their site frequency spectrums, the ratio of minor allele frequencies, and we see that when we break down those structural variants into whether or not they overlap with a gene, not at all or a little bit, the site frequency spectrum is fairly similar. It gets a bit more interesting when we consider that admittedly only about 20-25 cases where we have structural variants that encompass the entirety of a gene. And we can see that for these ones, there is an increase in some of the more moderate allele frequencies, which has really interesting implications for the individuals that contain these genetic variants. One of the reasons we're so interested in structural variants is because of their size. We consider that they might have a larger phenotypic impact on the organisms in which they occur, at least compared to single nucleotide polymorphisms. So this leads us to the question of, how do structural variants even work in terms of standing genetic variation? Here is an image that we're used to seeing very often when we think about selection. We've got genetic diversity, in our case, in the native range, and some of that is selected for, some of that is selected against under a new invasion or a new selection regime. And often when we think of this story, we're focusing on the the right side of the picture. Yes. And that's where all the interesting, cool stuff is happening, right? But one of the really big questions is, how does this even work? Why is there genetic diversity in the native range? Because selection's still happening there, right? And if there is a genetic variant that has a strong enough impact on a phenotype that it's going to be selected for or against in the invasive range, why is there not being some kind of effect on it in the native one then too? And is this not even more a concern for structural variants where their potential impact if they occur in regulatory or coding regions is even higher? Well, that's a very good question, and one way that people sort of have been starting to explore this idea is through the lens of balancing selection, which is the idea that there are certain selection regimes that may be capable of maintaining multiple different alleles within a population at moderate allele frequencies. And so we sought to investigate whether or not any kind of patterns hold true in balancing selection and whether they're the same across SNPs and structural variants. The way that we did this is we looked at outlier structural variants and outlier SNPs, things that were under selection in possibly the invasive range. And we used the underlying levels of balancing selection within specifically the native range in UK and compared them between groups. So between the SNPs that were classified either as red outliers or blue non-outliers, we saw that the balancing levels in the native range were, balancing selection levels in the native range were just a little bit higher, but not significantly so. And a similar story for the outlier structural variants. What we did find as a significant difference, though, was that different groups of variants had different underlying levels of balancing selection, which suggests that balancing selection may play an important role in maintaining genetic diversity in the native ranges, and that, more importantly, balancing selection will likely have a different impact depending on what type of genetic variant we're dealing with. So overall, we find that structural variants, or at least I found that structural variants were definitely a pain to work with, but conceptually very interesting to start digging down into, because there are so many assumptions that you just take for granted when it comes to working with SNPs that you kind of have to throw out the window when you're working with structural variants. But just because it's hard doesn't mean we shouldn't do it. In fact, I actually think it's even more so important that we start characterizing structural variant patterns where we can, because of so much noise in the data. Only through comparing multiple patterns across multiple species will we be able to start picking out the true trends that are sort of shining through. And with that, I'd like to thank you all and ask for any questions you may have.
Okay, great. Questions?
That was a really nice talk, thank you. Just a technical question about the balancing polymorphism beta that you had there, and how did you measure that?
Yeah, so luckily for me, that doesn't rely on structural variants at all, so I can pretend to be a bit more confident in that. So balancing selection, there's a really cool program called, I think, BetaScan, and it does high density SNPs. So you have to have whole genome resequencing SNPs, and what it does is it looks for regions where there is statistically an overabundance of SNPs at higher minor allele frequencies. And then, yeah, so to then assign some kind of balancing selection score to the SNPs or structural variants, I had sort of the underlying patterns in the genome of where there was high balancing selection, where there was low balancing selection in the native individuals, and I simply mapped the structural variants or the SNPs that were either under selection or not to those regions.
Right, so when you're talking about balancing selection, you mean within a specific population or across the whole? Within the specific UK population. So does balancing selection in the UK maintain genetic diversity, which then selection in the invasive ranges may act upon? Right, so you're counting the UK as one population in that also? Yeah, okay. Which, to be fair, I almost wish I could say maybe we shouldn't, but our UK samples were all from the same sample site and look like they're clustering pretty well. And so I think that's a fair assumption to make, at least of our data points. One day I'd like to have enough native range samples to say, no, we have to start considering population structure in the native range too, one day.
I obviously found that really fascinating. But I'm curious, kind of what is the range of sizes on the structural variants you're looking at? And did you notice, I don't know if you had enough data to parse apart you know, smaller versus larger, if you had a quite a big range of variants that you were
Yeah.
looking at. And if that played a role maybe in, you know, the different betas that you got for different types of structural variant, if you thought that might play a role as well.
Yeah, I can partially answer that question because some of these figures were just updated last night, and in the process of addressing reviewer comments, which has caused me to go from the top of this project down and change everything. So I've dug into the previous data set a lot, but some of the new trends I haven't. Yes, I've been teasing apart the across lengths, across types, across like repeat content and things like that. Largely because it's a short read data set, the structural variants are highly biased towards ones that are 10,000 or less. Vast majority of them are in the 1,000, up to 1,000 base pairs long. I'm pretty sure in the previous data set, which had fairly high overlap with this renewed one, quite a few of the inversions were a little bit longer. That being said, the way that we got the balancing selection scores, where we actually used SNPs directly upstream of the structural variant. This touches on some of the technical difficulties and the things that were really annoying to deal with structural variants, because often you go through and with SNPs, you just consider direct overlap because it's one nucleotide base pair. And I was halfway through doing that before I realized I was considering overlap with a deletion, and that doesn't work because it's not even there. So in order to then do balancing selection for the structural variants themselves, I focused on the closest SNPs that were upstream to the sort of first break and start point.